Hey guys. Okay, so we're here to talk about specifically signal transduction in this section. Okay. So signal transduction pathways kind of influence how the cell's going to respond. So it's like the game of telephone. The more people involved, the more steps involved, the different message is going to be passed on, whether we're emphasizing it or breaking it down, things like that. And so it's kind of the same concept here, that depending on how we're moving this message will kind of influence how the cell's going to respond. So they can change gene expression if it goes in and directly affects the DNA. It may change the function because it might lead to the creation of certain types of enzymes or things like that. So it can alter phenotypes. So instead of having red hair, maybe it makes it turn blonde. I mean, if you get this kind of change in message, you find this, especially think about the snowshoe hares, where in the colder weather, they're white, and in the warmer weather, they're brown. So this is one of those situations that a signal transduction pathway can actually help affect that, or it can ultimately lead to the death of the cell. So if to change these pathways, there are mutations. We know that mutations occur, and if a mutation happens to, for example, the receptor protein or any component of that pathway, anywhere along the pathway, there's a mutation that's going to change the transduction or the movement of the signal. So here's a little practice-free response for you. Some diseases, such as cancer and diabetes, are caused by defective protein phosphatase. A, explain how such a defective protein would affect a signal transduction pathway. Okay, so obviously receptors are very important. In eukaryotic organisms, we have two main categories for those cell membrane receptors. We have the G protein coupled receptor. One of the groups did that. And we have ion channels, which is what another group did. So looking at our GPCRs, our G protein coupled receptors, the majority, so the largest category, the majority of the surface receptors are actually going to be GPCRs. And they're really important in terms of the sensory system. So what happens is a G protein that it binds to a G protein that can then bind to GTP, which it's similar to ATP in terms of the energy aspect. So that's where it comes into play, except the key nucleic acid involved is different. It's, if you think about when we talk about DNA, we talk about the ACTG. So ATP uses the A, GTP is going to use the G. Okay, so GPCR, the enzyme, and the G protein are all totally inactive until that ligand, that chemical messenger, binds to the GPCR. So that's on the extracellular side, outside of the cell. So it's all inactivated till that ligand binds. When the ligand binds, that sets off the reaction. So the ligand binds, which then causes the cytoplasmic side, one inside the cell, to change shape, therefore activating it. This activation allows the G protein that we changed the shape, right? It allows that G protein to bind to the GPCR. So now the G protein is bound to this GPCR molecule. That activates the GPCR and the G protein. So this is now active. And in this process, GDP becomes GTP. Just like when we go from ADP to ATP, it's the same process, that same phosphorylation process. Okay. So then the part of the activated G protein can then bind to the enzyme. So we've released that little activated G protein and it comes over and binds to the enzyme. So now we have a nice active enzyme. And that enzyme can then amplify the signal. So think of it in terms of we went from one signal, now we have 3 signals if you look at the diagram. So we made it stronger, therefore leading to the cellular response. The other biggie is the ion channels, and these are ligand-gated channels. So really 2 of the ones that y'all did with your modeling were ligand-gated channels. It's just one of them was very specific versus the other one was generalized. Okay, so ligand-gated channels, they're going to be located within the plasma membrane. And in particular, these are really important when it comes to the nervous system. One of the key factors that allows a neural impulse to move down a nerve. The receptor acts as the gate for the ions. And so when the ligand binds to the receptor, the gate opens or closes depending on what direction and what ions it needs to do, allowing for the diffusion of certain specific ions, for example, sodium or potassium. This initiates a series of events that leads to the cellular response. So, for example, with a nerve impulse, we have to have kind of we build a gradient, so we put more positives outside, less inside, and then to make the neural impulse move, we drop and balance it back out, and then we reset the system. So we're moving these ions. So the ligand comes in, attaches to the channel. In this diagram, it causes the channel to open, and that open channel is allowing the neurotransmitter to move in, and it's allowing ions to move also. So we've got sodium and calcium going one way, we have potassium going the other. So we're making this concentration gradient concept. Okay, so hopefully that helps y'all out, and I will see you in the next lesson.